Hi, so I'm going to get started. My name is Matt Lebo. I'm from the Department of Personalized Medicine, the Lab for Molecular Medicine here, as well as Green Women's Hospital and Harvard Medical School. I'm going to talk today a little bit about genome and exome sequencing and our experience with it as a clinical laboratory. So really the way that exome genome sequencing can be thought of is really a component of the larger landscape. So if we think about it, gene panels are very small, usually around 50 to 100 genes, a very small component of the overall genome. The medical exome, which I'll mention in a little bit, is sort of what the clinically relevant genes are that we have. And that's probably more in the order of 4 to 5,000 genes, maybe about 10 to 12 million base pairs. The full exome, as everyone knows, is closer to 60 million bases, 22,000 genes, and then the entire genome, about 3 billion bases, about 5% of which is highly conserved, and 2% which is the coding exons. And if we think about sort of the testing landscape today, that really is separated into sort of panel testing and exome and genome testing. And so panel testing is really clinically well-defined genes, and what we aim for there is really high coverage and completeness. And so we're interested in completely covering every single base within our defined ROI. Exome and genome sequencing is a little different. It's a lower coverage, obviously. The completeness is less than 100%, and it's not usually filled in. And it's really, right now, for more diagnostic odysseys or complex overlapping phenotype stuff. What I'm here to talk to you a little bit about is when you're thinking about exome and genome sequencing, it really is, in the standpoint of development and testing, very similar to NGS panels. So if you think about the testing workflow really is the same regardless of which of the methods that you're doing. So genetic tests will come in, there'll be accessioning of that test, the wet lab case management portion of it. For pretty much all of today's tests, there is some bioinformatics components. Even Sanger sequencing traditionally today has bioinformatics pipelines to help automate that process. There's curation of the data, determine what sort of is pathogenic, what will end up on the report, what won't. There's drafting of the report, and then the sign out and report delivery. And really, again, this is the same regardless of whether you're doing exome, genome sequencing, or clinical targeted panels. And similarly, the test development stage is actually the same whether you're doing exome or genome sequencing. So the way that we tend to think of test development is there really is some development where there's an optimization stage of it, where you're really learning your assay, learning what you're doing, figuring out what the appropriate analyses are, and actually understanding sort of what the strategy for that testing will be and doing some optimization of that whole process. After that, you really then get into the validation stage. I'll talk a little bit about our validation of our exome and genome in a couple slides, but really what you're looking for is sort of the reproducibility, the robustness, and how accurate your assay is. It's important here to sort of include different variant types, so being able to understand how well you can detect indel substitutions, CNVs if that's what your assay is actually doing, or actually understanding sort of common pathogenic variants and your ability to do that if you're more focused on sort of a genotyping type pathway. But again, this is sort of the similar process for exome and genome sequencing as it is for clinical targeted panels. It's just sort of a broader scope of the testing you're doing, and maybe even a limited set of samples that you can work on due to the cost of actually doing the sequencing. So again, the exome and genome development, again, is similar to the panel testing. One of the things that I want to point out is that traditionally labs sort of bundled their validation process from beginning to end, but it's really becoming, more recently, it's becoming evident that you can actually separate the technical validation from sort of some of the more downstream bioinformatics validation. By technical validation, I mean sort of how well the platform is performing and the reproducibility, both inter and intra-run variability of the platform, and really how well that platform can produce what you're expecting it to produce. And then on the separate end is the bioinformatics component, where if you're taking that data and actually doing some of the alignment and variant calling, coverage, annotation, filtration, whatever you may be doing for your downstream steps, and actually validating that whole process and the ability to produce results at the end. And you can actually sort of decouple these two things, and the ability to actually have this decoupling enables you to sort of upgrade one component versus the other component without having to do sort of this full validation of everything. So it's really important to start thinking about this when you're implementing it, that you can sort of do again, this technical validation versus this more the further downstream bioinformatics validation. I'll talk to you probably a little bit more on the bioinformatics validation side today than the technical side, but it's when you think about how to validate the platform, there are specific things that would go into that as well. So one of the things that I'm going to talk about again in our talk is in our exome data, we've worked to sort of produce a more clinically relevant exome, and I'm going to be mentioning the words medical exome, and I just wanted to sort of let you guys know what that was. So really the medical exome was a collaboration between Birgit Funk, one of the directors at our laboratory, as well as her colleagues at Emory and CHOP. And the goal of the medical exome initiative that they put together was to really define a set of clinically relevant genes and then to enhance sort of exome content to make sure those genes were really well captured and really well covered. So oftentimes there's sort of specific exons that aren't captured, there's specific genes that really weren't part of the capture kit or specific transcripts that weren't part of the capture kit. The goal was to sort of look at what was underperforming and then boost those regions to really get higher completeness and higher coverage of the clinically relevant genes, which is going to be very useful in sort of clinical exome sequencing. And so this is sort of a pilot that we use the results from to sort of determine what, or to use this enhanced exome kit to really drive our further processes. And I will sort of mention a little bit of the results from this, although I won't show the difference between the older and newer one, but we were able to actually get much higher coverage and much higher completeness for the clinically relevant genes using this approach. So when you're going through your, again, exome and genome pipeline and even some of your NGS pipelines, your first step is from the bioinformatics components to the alignment, variant calling, and annotation processes. And so just as a brief overview of what we're doing, for our genome cases, we're using a modified BWA and GATK approach to really parallelize this and to reduce the computation time down to something that is a little bit more manageable. And just to give you guys a rough size of what we're doing, we're using 600 million paired end reads. It's about an average depth of 40x, and it's about 95% completeness over the entire genome. Our exome pipeline is a little bit more standard since the processing time and compute time is a little less. So for here, we have about 70 million paired end reads. Average depth is at least 150x, so usually a little bit higher. And because of this medical exome initiative that we did of enhanced coverage there, we're actually getting closer to 200x for disease panels when we're looking at known disease panels that have clinical relevance. And our current pipelines identify right now SNVs and small indels, although we are working on including CNVs and other structural variant calls in addition. But for SNVs and small indels, we're getting about 5 million variant calls pre-quality filtration for our genome, and about 120,000 variant calls pre-quality filtration for our exome. So it is a large number of variants that we're sort of sifting through, and I'll talk a little bit about the filtration a little bit later on in the talk. So when you think about how to validate this, there's actually a few different good sources out there for validation. And again, validation of exome and genome sequencing, although it does fit the mold of sort of any laboratory developed test, there are some caveats. Because of the cost of sequencing, you can't really do the breadth of individuals that you might want to. And also, you can't necessarily validate all those variants that you're finding if you do the breadth of individuals. And so really, a lot of the way that exome and genome validation is done is focusing on specific samples and understanding the accuracy across that sample in multiple different regions. And so the sample that most people tend to use today is NA12878 from the Coriell samples. And so there's a few different initiatives that have tried to look at what are really well-known variants and well-carried variants from this sample. So the first is the GetRM database from the CDC, and what this includes is variant calls that were confirmed via orthogonal technology, oftentimes Sanger sequencing, by submitting laboratories. And most of the laboratories that submitted to the GetRM project were clinical laboratories that really did look at confirmation of these variants across multiple different technologies. And we actually use a set that we did upload to the GetRM project, and the one that I'm going to talk about today for our laboratory is about a little less than 450 Sanger variants, or variants for NA12878 that we've confirmed with Sanger sequencing. And so we actually know both all the places that we have variant calls and all the places where we have homozygous reference. We can get an understanding of both sensitivity and specificity for these regions. The other initiative that is really well established is the Genome in a Bottle from NIST. And really, this is looking at integrated variant calls from multiple different technologies, multiple different platforms. And what they've been able to do is create a high confidence set of variant calls. And we've actually tested this on our data, and it actually performs very well in comparison with our Sanger data. And they have a much broader region, obviously, than the limited region we have with Sanger follow-up. And this is another great tool or a great data set to be able to actually start thinking about sensitivity and specificity measures for your exome and genome sequencing. And then finally, GCAT is sort of a website that uses the NIST data set, so you can sort of automate the process of doing some of the analyses if you're interested in doing that. So this is the validation results of our genome sequencing, and our exome sequencing was pretty much the same. Really, we had, again, a little over 400, a little less than 450 Sanger confirmed variants across about 700 KB of sequence. And based upon our genome sequencing and our exome sequencing, we were able to identify all of those. I think in our exome sequencing, we missed one variant that was in STRC, which is one of the most highly repetitive genomes in the genome, although we were able to call 2 other variants in that one as well. And so we actually are getting very good accuracy and results from this data. And actually, if we think about the specificity, we're actually not getting that many false positives. And so we initially had a fair number of false positives when we looked at it, but most of those had really poor quality. And so by establishing specific quality metrics, in this case, we use Fisher strand bias and the quality by depth measures from GATK, we're actually really able to reduce the number of false positives that would end up in our final data set. And we again get very similar results with our exome data, and we're actually able to use the same thresholds for our exome data that we're using for our genome data. So it shows that these thresholds are actually fairly robust for the platform and for the tools and softwares that we're using. Finally, another way you can sort of think about how well your data is performing is to do concordancy checks with other data sets. And so the 1,000 genomes data isn't necessarily a gold standard data set, but if you compare your results to something like 1,000 genomes data, you can get an idea of how well correlated you are. And obviously, the higher concordance rate between the 2 data sets, the more confidence you might have in sort of what you were able to do. And so we actually had fairly high concordance, especially in SNVs. And this is both sort of just general concordance, was the variant called, and also what was the zygosity of that call. So we were able to get concordance levels of close to 100%, at least 98, 99% for both of those, although it's a little bit lower for indels again. So again, we also were able to do this comparison to the NIST high quality data set. And so what we're looking at here is our sensitivity and our positive predictive value analyses against the NIST data set. And specifically, we're looking at the entire exome versus just this medical exome of about 4,000 to 5,000 genes. You can see we actually get very similar results in terms of our sensitivity analysis and our positive predictive value. And you can see that this is another great way to think about how you're going to validate your data set by using this tool. And it really does give you the ability to, from a much broader standpoint, across a much wider range of genes than maybe you have data for, to really be able to start doing the comparisons to get the accuracy of your data and of your technology. So sort of this was the validation of the platforms, and one of the things we do in addition to sort of understanding how well we're calling variants, understanding how complete our assay is. And so one thing that's also important to do is sort of a coverage analysis. So this is a little bit more than just sort of what's the average coverage across your region. It's really understanding how well things are covered. So for instance, you might think about the base level coverage, and so one of the metrics that we look at is the percent of bases across certain regions that are callable, so above your thresholds, that have low coverage, that have 0 coverage, or that have poor mapping quality, and so other different thresholds may go into this as well. You can also think about the exon level coverage, and so traditionally people tend to think of genes sort of how well covered they are at certain amplicons, at certain exons, and so we tend to think of, for example, how the percent of the exon that is at a callable level. So how many exons do we have where 100% of the exon is at 20x for exome or 8x for genome, that sort of measure? Or how many of them are at like 75% of the bases in that exon are at a callable level? You can also think about gene level coverage. And so from a gene level coverage perspective, it's really the percent of gene that is fully covered, and so you know you have really good data, the percent of genes that are partially covered, and you can break the partial coverage down into different metrics, or the percent of genes that are not covered at all that you know that you're never going to get data for and that you're going to have really trouble with in terms of your clinical assay. And really, you can do this coverage analysis across multiple different ROIs, and I'll show you in the next slide that we've done this across different sort of exome ROIs and as well as disease subpanels. So this is an example of our coverage data. So here we're looking at the exon level coverage data. Sorry that might not be presenting that well for you guys, but really what we're looking at is the percent of exons within these different regions of interest that are fully covered at 20x coverage for our exome data, or at lower coverage. So the dark blue is the exons that are fully covered, 100% covered at 20x. You can see that if we look at our ROI, the first thing we're looking at here is the exome plus or minus 15 bases. The second one is the full exome plus or minus 2 bases. Then the third one is the medical exome, which is, again, the region that we enhance coverage for. And then the following 5 are sort of more disease subpanels, including ACMG and sentinel findings list. You can see here, even for the exome data, we're getting pretty complete coverage across most exons. We're getting complete coverage across most exons at greater than 80%, 90% level for our full exome. And as we go to the things that are more clinically relevant, this is really hovering closer to 97, 98%. You can see that it makes it feasible and it makes it much more complete to think about actually doing more targeted panel sequencing actually off of exome data. So that's sort of the validation process of exome and genome sequencing. And the next step in the process is really to think about what you do once you have that data. And so once you have sort of variant calls that you trust and that you believe are real, the way that we approach this is to do a really broad annotation of variants. And there's multiple different software tools that are available. I've listed a few here if anyone's interested. We use a few of these tools and some of our own in-house tools to annotate variants with a greater than 100 different variant features, we'll call them. These features can include nomenclature, the type of variant, functional prediction tools, conservation, frequency, mitotic frequency, that sort of thing. And also presence or absence in databases and what the databases might say with them. So we also annotate, for instance, using ClinVar. We then upload all of these patient variants and the annotated variants to our internal genomic data repository, which enables us to sort of filter based upon quality metrics on patient variants, on different annotations, to really produce a smaller subset of variants that we would look at during our exome and genome sequencing process. So one of the things that's important to consider when doing exome and genome sequencing is that most of the time you'll have someone who comes in with some sort of phenotype. And one of the first things you do, especially if you have singletons and don't have parents, is you might need to limit it based on really what their disease is. And so one of the first things to do is actually try to identify sort of these gene lists that are phenotype driven. And so there's some issues here. There's really not that great ontology available, and there's not great sort of gene disease associations that are really well curated. And so in the future, there'll be a lot more tools out there that enable you to sort of do this. But right now, it's a little bit harder to sort of create these phenotype-driven gene lists, and it's oftentimes a little bit more manual. There are a few sort of software tools out there that can help you with this. So there are some ways to sort of capture structured phenotype data. So Phenotypes, PhenoDB, and SignalConsult all sort of do this. And then there's tools like Exomiser that can sort of help you take this and select specific genes of interest. Although there's a lot more work that needs to be done in this area to sort of automate this process. But this is one of the key components, and right now is probably a key time component as well, is to really determine which genes, if you have a sort of phenotype-driven gene list, that you're going to focus on in order to do the filtration. So again, once you have sort of your phenotype or your gene strategy that you want to do, or you maybe have a genetic strategy, so you look for de novo variants or compound heterozygous, or you're looking for sort of homozygous variants for consanguineous families. Once you sort of have that strategy established, then you can do this filtering to get to a sort of smaller set of variants that you want to manually review. Again, we do this sort of against our genomic data repository. We do this using SQL queries right now, although we're hoping to sort of make this into much more UI friendly for our geneticists and GCs. And there are sort of multiple software solutions that do work in this space. Some of them do both annotation and filtration. And I've listed a subset of them there that could provide ability for someone to be able to do this in the future, or do this now, I should say, but in their own laboratory. So once you have the list of variants that are really filtered down, it really is important to dive into these and actually do a thorough assessment of this. And so really, we do a manual assessment of each of the variants that end up in our filtered list, and we dive down for the ones that are most likely clinically relevant into a much more expanded variant assessment tool, which I'll talk about next. And one of the things that's important to consider when you're doing this filtration, which is a little bit unique to genome and exome sequencing and a little different than traditional target panels, is you actually then have to do a gene assessment often to really determine how good the evidence is for this gene-disease association. And so one of the additional components here is we have a process that we have in-house, but it still is another time component where you have to think about, is this gene and this disease association really well established, or is it really just one family that was mentioned once in the literature a long time ago? And so really, how likely is it that even if the variant seems to be pathogenic, that it really would be pathogenic for a specific disease? So again, we mentioned we have a variant assessment tool that we use in-house that can be made available to others if they're interested, but really the goal for us was to produce a very robust, well-validated way to clinically interpret our variants. And so we have an Excel-based tool that we use that takes in a lot of annotated information, presents it in a way for fellows and GCs to really look at an interactive and integrated view, enables us to really do this in-depth dive into the variants that we think might have the most clinical relevance. This is an example of a screenshot from it, as well as a paper that we published last year that sort of talks a little bit about it. Again, this is sort of one page of multiple tabs that we look at. This is the conservation tab to understand sort of from the functional prediction tools how well conserved or how likely it is to be deleterious. And then we sort of present this again in a summary view where we then determine sort of our evidence section that would go onto a report. So this is the example of the evidence section, or the evidence that we associate with the variant that we would actually add on the end of that we add onto one of our reports. And this process is actually very important. So if you think about variants that have been determined to be pathogenic or not in the database, these are the variants that have been determined to be that were called pathogenic previously in a database, and after we did thorough evaluation of them, what actually was the pathogenicity of them. And it turns out that only about 3% of variants that were called pathogenic in some of these databases did, after manual review, did we feel like actually met the pathogenicity criteria to be included on a report. So again, it's very important to go and dive a little bit more deeply into a lot of these variants, because the databases that are currently available really aren't as high quality as they hopefully will be. So again, this is sort of quickly what is what we do. So we look at the variant annotations, we use a bunch of stuff to do our variant classification. And for genome and exome sequencing, it's actually a little different. We only report likely pathogenic and pathogenic variants. And then occasionally we will report a small set of VUSs that really seem relevant for this individual patient, but might not be for another patient. So that's the small set of variants that we end up reporting on our clinical reports. And this is an example of sort of what our clinical exome report is. We try to create a one-page summary that puts in all the relevant information, including the variants that are relevant to the disease and any sort of incidental findings that we've identified. And then in subsequent pages, we do the more detailed information of really why we classify this variant in that manner, and really what the disease information for this individual variant is. So this is sort of a way to sort of present a one-page view of the overall results, and then further go into a lot more detail if the clinician wants to understand sort of why we came to these conclusions in the beginning. And this is something that we've developed sort of in context with the MedSeq project, which I'll talk about next, but to give you an idea of sort of what we aim to give back to clinicians so they can actually have an understanding in a much more simplified manner of what the findings that we found were. One of the things we've also been adding is sort of a supplementary coverage report. And so if someone came in for a specific phenotype and we really did one of these phenotype-driven gene list analyses, it's really important to be able to let that clinician know how well things were covered or not covered. So that way, in case there was a gene that is really relevant to disease that was not covered, we can flag that and sort of let them know that although we did look for this subset of genes, this one critical gene we weren't able to cover, and so you may need to do additional follow-up testing if you still suspect that this sort of gene may be responsible for this patient's disease. So we've been using our exome and genome sequencing, obviously clinically, but also to support a few different clinical trials. And so I'm going to go through quickly the couple different clinical trials that we're really looking at. So the first one is the MedSeq project, which is a randomized controlled project where we're looking with patients with disease, specifically in this manner, cardiomyopathy, that are presenting to cardiologists, as well as healthy individuals that are just coming into their regular primary care physician for routine care. And we do sort of a split between people who get standard care and people who get standard care plus a genome report. We're then looking at the outcomes from, we're delivering back the genome report, and we're looking at the outcomes, including medical review, for the people who got genome sequencing and people who got regular standard of care for both the healthy arm and the disease arm. And for this one, again, we're using a filtering strategy where we're looking at just incidental findings, but it's a really large incidental findings list. So it's all, we initially look at all things that have been previously determined to be pathogenic using HGMD or ClinVar or our own internal database, or loss of function variants across the medical exome that we then go and review. We also return pharmacogenomic variants and blood groups information in the context of the MedSeq project. And then sort of look at, again, how the clinicians deliver this information, how the patients respond to the information. And actually, if we think about it, it's even though we have a really large expanded incidental findings list, we're really not finding that much. So we've done about 40 of these cases so far, but for the first 7 cases that I present here, we actually only find 1 to 3 variants, 1 to 3 incidental finding variants per individual. And most of these tend to be carrier variants. Very few of them sort of tend to be dominant Mendelian disorders that we're finding. And so it really is a very small amount of variants that end up being returned, and a much smaller amount that we actually initially expected from the onset. The other project that we're supporting is the BabySeq project, which is again doing a similar project, specifically looking at sequencing and newborn sequencing sort of in the context of genome sequencing. And so what we're doing here is we're again doing a randomized control project that we're looking at NICU infants that are randomized to either receive the genome sequencing or standard care, and again healthy newborns that are either going to do standard of care or genome sequencing. And so this is a project that is just getting started now. Hopefully we'll have some results in the near future to be able to present, but it's something we're really interested in pursuing along with our clinical assays, is how we can understand really the effects of genome and exome sequencing in a clinical context by doing these sort of randomized control studies. So with that, I'd like to sort of thank a lot of the people that I work with here at Partners Personalized Medicine. And I think with a few minutes left, we can take any questions that you guys can submit through the Q&A session. So thank you for listening. All right, so I guess I don't have any questions coming in, but if you guys think of any questions as your follow-up and you want to email me, feel free. It's just mlebo@partners.org. I'd be happy to sort of share some of the tools that we've developed or talk to you about our processes and our experiences here at Partners Personalized Medicine in establishing sort of exome and genome sequencing in a clinical context. So. All right, thank you.